Hello everyone. Thank you for joining us today. See some people coming in at this moment. Uh, Please, for us, a first to use the GoToWebinar platform for a webinar of JC Discovery. So we're very happy to welcome you. Okay. So like I said, thank you for joining us today. My name is Quentin. I'm business developer for JC Discovery, and it's my great pleasure to introduce you to Dr. Bernard Malissen for his talk on harnessing the clinical potential of adaptive T-cell therapy through innovative engineering and preclinical mouse models. Hello, Bernard. Can you hear me?
Yes. Hello,
Great.
Quentin.
Great. Hi, Bernard. So Bernard is Chief Scientific Officer at JC Discovery and has over 40 years of experiences in elucidating the fascinating mechanisms that are present in T cells, albeit mouse or human T cells in a healthy or disease context. And throughout his career, Bernard has received numerous awards for his research in immunology and consistently ranked amongst the most highly cited researchers in the field. So in 2012, he founded the Center of Immunophenomics based in Marseille, which provided the scientific community with valuable resources like preclinical mouse models and an expertise in immunophenotyping. And today he's going to share with you his experiences in studying T cells and how this can be translated into the development of, amongst others, CAR T cells. So to just secure everyone's connection, I would say, shut down my camera so we can focus really on the slides. So, okay. So, okay. We can go to the next slide, please. Yeah. So as a side note, if you're encountering any technical issues or have a question in mind during the talk of Bernard, please contact us through the small gray question mark on the right side of your screen. This is the same tool you will use in the Q&A session just after the presentation of Bernard. So I'll take a few more minutes before giving the word to Bernard to present you JC Discovery. So JC Discovery in the next slide, please. So JC Discovery is part of the Janvier Group Biosciences, along with 9 other expert companies offering a unique combination of products and solutions, which we divide typically in 3 branches. So the first branch is the models and the associated solutions, including mouse models and organoids. The second branch is focused on the drug discovery and preclinical services with experts in approaches like small molecules, monoclonal antibodies, T-cell therapy in the case of JC Discovery, in therapeutic areas, amongst others, oncology, autoimmunity, but also cardiology, metabolic disorders, or even liver-related diseases. And then the last branch is the bioproduction branch, with the engineering and the biomanufacturing of biological reagents and raw materials, such as plasmids, antibodies, small molecules, and so on. So please don't hesitate to visit our website, janvier-group.com, if you want more information on the preclinical solutions that the group can offer. So next slide, please. So JC Discovery is based in the south of France, in Marseille, and has been founded in 2022 as an initiative between Janvier Labs, a family business active in the breeding and distribution of rodent research models, and then on the other hand, the Center of Immunophenomics, which I briefly mentioned before. So next slide. So small word on its founders. We have Thomas Janvier himself as our CEO, Bernard Malissen, as mentioned before, operating as our CSO, and we can count on the support of Anna, Frédéric, Hervé for their scientific advice on the projects we're collaborating on with our partners. Next slide. So here you have a summary of the team focused on the actual experiments that we perform for our partners, ranging from study directors, engineers, and technicians. And If you ever recognize the faces of, for example, Anaïs, Javier, and myself, we will recently also be present on conferences focused not only on cell therapies, but also the AACR in Chicago in April. So don't hesitate to stop us and to have a chat with us related to the science that we're all doing. So next slide. So from my side, I'll usually be your main point of contact, um, in particular to have a first evaluation of your requests and facilitate the collaboration process. If after this talk you're interested in contacting me for services as described by Bernard, please do so using the mail or telephone number described here. So next slide. So I promise, give me another minute and then I hand over the words to Bernard. So I want to share with you a very short summary on JC Discovery's expertise and preclinical solutions. So typically we help our partners to identify, validate, and prioritize new therapeutics aiming at or activating or dampening the immune system in the field of immuno-oncology, inflammation, autoimmune, and infectious diseases. And we do so by performing scientific experiments on validated models with a strong focus on using standardized protocols, allowing us to analyze and compare data over time. So ultimately, a good interpretation of scientific data allows you to elucidate the mode of action of your drug candidate at the molecular, cellular, and systemic level. So next slide. So JC Discovery has three departments, so everything is divided in three apparently. So three departments with its own expertise inherited from the Center of Immunophenomics. So the first one is focused on the generation of genetically engineered mouse models. So think about knock-in, knock-out mouse models using different tools. The second department is responsible for the use of our proprietary predictive mouse models and immunophenotyping solutions. And then finally, the department most important for this talk today is dedicated to support cell therapy developers in their preclinical research. And that's also the department led by Bernard and Javier, which is present in the background next to Bernard, but I'm happy he was able to join. So I hope I was able to give you a better view on our solutions, and I think I'm ready to officially start this webinar. So Bernard, the floor is yours.
Okay, thank you, Quentin. So I will divide my presentation in two. The first one will be very brief, and essentially I'll give you some outline of the scientific service, technological services that we are providing at JC Discovery. And the second part will be to try to show you how innovative engineering approaches and preclinical mouse model can be contributing to harness the potential of adoptive T-cell therapy. So very rapidly, JC Discovery, the kind of services we are offering to our customers, it concerns, for instance, CAR T-cell design and production, and also in vitro and in vivo evaluation of the function and the efficacy of these CAR T-cells. And we have also another part of our work we capitalized on our knowledge on interactomics. And through interactomics, we can provide interesting information on the mechanism of action of drugs and biologics, which are intended to modify or enhance therapeutic T-cell efficacy. So for instance, for CAR design, I mean, we are capable of designing any kind of formats, I mean, from regular CAR to TCR-T to ITSTAR-like formats. And we have a rather wide experience on engineering multicistronic vectors with large payloads. And so once the CAR has been or the different formats have been constructed after we integrate them for sure in the genome of primary human T cells, and we do so like many others using lentiviral-based gene delivery or doing knock-in through CRISPR-Cas9 based model. And at the same time, we are introducing the CAR or the EAT. We can simultaneously modify the genome of the primary T cells through knock-in or knockout. And these lead to a kind of pipeline, which is a very classical pipeline for people trying to design and analyze the function of CAR T cells. So we have a GMP compliant process that permits to obtain, let's say in 12 days, a sufficient number of CAR T cells to infuse large cohort of tumor-bearing NSJ or NXJ mice. And then, based on that, we can perform some regular in vivo stress test, and the readout again is very classical for sure under survival, but also we use EV spectrum imaging system for tracking the evolution of the tumors. So just a couple of slides on interactomics, and just give you two examples how we can use it to prioritize human targets or drug. That's just an example. We published a long time ago in Journal of Experimental Medicine, where we have a compound that we call X, and we wanted to know where this compound was acting inside the T-cell receptor signal network, which is, as you know, it's a very complicated one. And in fact, by using the interactomic tools, in less than 4 months, I mean, we managed to figure out that this compound was acting extremely upstream in the cascade of signalization that is operated by the T-cell receptor, and essentially preventing the information from flowing from the LCK protein tyrosine kinase to the SLEEP76 adapter. So that's just one example. Another example of interactomics is to help designing combination of antagonist or agonist antibody in immuno-oncology. And again, I take an example that was worked out by Javier, which as you are aware, there is the PD-1 molecule, which is very famous, but there is another molecule called BTLA, which is a close evolutionary relative of PD-1. And people were thinking that they are sharing a lot of signal transduction component. And using quantitative interactomics, what we showed is that PD-1 is most specifically working with the SHIP1 protein tyrosine phosphatase, and BTLA with another member of this family called SHIP2, which is very interesting, but it's just a quantitative aspect. I mean, SHIP2 can also work with PD-1 when you kick out SHIP1, and vice versa. If you kill SHIP2, SHIP1 can work out. So there is some strong redundancy in between these two effector molecules, meaning that if you develop a drug which is blocking, for instance, SHIP2, it will be totally non-efficient for preventing the function of PTLA and PD-1, because SHIP1 will be taking over. So we touch with that all the issues related to redundancy, which are extremely important to take into consideration when you want to devise a combination of checkpoint inhibitor, for instance. So up to now, I mean, we have a recent company, but we have been able to work with Amgen Horizon, Altrubio, Galderma, ICB Tech, Q10 32 Bio, and Oz Immunotherapeutics. And also through the fact that we belong to the Janvier group, we can also use in a very efficient manner humanized mice, or through the Edelris partner, eventually devise PROTAC or protein degrader for controlling the signaling in CAR T cells. And also very importantly, MIMABs, which is located in the same place as us, has the capacity of developing through beacon screening some T-cell-like antibodies, which allowed us to arm our CAR with proprietary molecules. So let's move to the main part of my talk, which is essentially to try to figure out if the knowledge we are learning from the study of T-cell signaling pathway can be used to make better CAR T-cells and therapeutic T-cells. In other way, can we move from some kind of tinkering to rational design? And as you are well aware, I mean, T-cells, they are absolutely beautiful biological objects with a lot of surface receptors, which are displayed by the T-cells, and essentially they're intended to sense the information which are conveyed primarily by dendritic cells. And the beauty of the T cell is that they manage to compute all this information to make some informed decision. And for instance, when the T cell is stimulated in some kind of acute antigenic stimulation, you have a very classical unfolding of events which is occurring. I mean, the naive T cells expand and also they become polarized, functionally speaking, and then you end up with effector T cells and memory T cells. This kind of paradigm is a little bit different in the case of tumors and of T cells which are subjected to chronic antigenic stimulation. As you are aware, the cascade of differentiation of the T cells, you go from naive T cells, but then after it's a little bit different. You end up with what is called T precursor exhausted cells, which has some kind of stem cellness property. And once they reach the tumors, that's the one which give rise to the exhausted cells, which are capable of destroying the tumors, but will have a very short life. And essentially, if this kind of flow from here to here is not functionally properly, essentially because at the level of the tumor, the dendritic cells will not be doing their job. So then the tumor is taking over. So all these business, we have to remember, start from the discovery of Zeli Geshar at the Weizmann Institute in Israel in the late 80s. He worked out absolutely beautiful format of chimeric molecule where essentially he was using part of an immunoglobulin, the single chain heavy, which is conferring the specificity of the chimeric antigen receptor, and he was docking it inside the membrane of a T cells through the hinge of a molecule called CD28 and also the transmembrane segment. And then in a very smart way, he was introducing inside some component that will be converting the binding of the single chain heavy to an antigen to the fact that you would be turning on the activation of the T cells. And originally people were using only the CD3 zeta, which is a component of the T cell receptor CD3 complex, but then they added CD28 co-stimulatory module and many other modules. So it's a very beautiful architecture, but we have to keep in mind that at the beginning it was assumed that when you plug a car inside T cells, essentially it will deliver exactly the same type of signals like the T cell receptor is doing. But we have to revise more recently these views because it turned out that the car are poorly connected to the let's say to this pathway, and there was very beautiful work from the group of UPA in Austria who showed that, and more recently there was also data from people using mass spectrometry like us, Interactomics, where they show that in fact the coupling of these intracytoplasmic domain to the downstream pathway is not as efficient as we thought. There is another issue that has been popping out, is that the fact that you fit one signaling motif there and another one there, it's a bit of tinkering, and it's not exactly what's going on at the level of the T cell receptor. And a couple of papers also showed that these stimulatory domain, they don't sit exactly in their natural membrane proximal location, and it has also some impact on the efficacy of the CAR. So to try to substitute to this kind of issues, which made the CAR not as strong as we'd expected, there's been an unbelievable amount of work that has been done to boost the activity of the CAR T cells. And essentially there was two ways. As you know, the T cell receptor is a very important molecule on T cells, but it receives some help from co-stimulators, such as the CD28 molecules. And also, I mean, there is some molecules which are down-regulating the function of the T cell receptor, like the PD-1 co-inhibitor. So it's very evident, it's very simple. So essentially, if you want to boost the property of the CAR, you can try to accelerate the way the co-stimulators are functioning, essentially by using gain-of-function mutation, but we'll not be discussing that today. But there is a more classical approach, which is to try to eliminate from the CAR T cells the brake which are stopping the best function of the CAR. And there is an incredible amount of work which has been done. For instance, people doing some fundamental research in T cell activation, they knew from a very long time that there is molecules such as the CBL-B molecule, which is coupled to the CD5 molecules, which are in fact a module which is desensitizing the function of the T cell receptor and of the signaling pathway. So based on that finding, there's been a huge number of papers where people were deleting CD5 or deleting CBL-B, and in fact, it turned out it was what was expected. It led to some enhancement of the anti-tumor activity of the CAR T cells. And dozens of experiments have been done. People have been targeting CTLA4, PD-1, RAGNAS, ROCK1, CISH, BTLA, and so on. And most of these papers, in fact, confirmed that the molecule that they were using were indeed some break, because most of them were enhancing the function of their CAR. And there is a huge amount of work also which have been devoted to introduce some mutation in the CAR T to delete genes that may render the CAR T cell dysfunctional and exhausted when they are entering the hostile tumor microenvironment. And in parallel, some experiments have been done for enhancing the metabolic fitness of the CAR T inside the tumor microenvironment. And there's some very exciting paper. For instance, this paper came out last year in Journal of Experimental Medicine. They claim that they have almost immortal-like T cells by changing some transcription factor. And I just want to show also these experiments coming from Sebastian Migorena group, which are changing the epigenetic substrate which are modified in these cells, and that also lead to some enhancement. So more recently, people have went one step further. They say, okay, that's fine. Up to now, we have been taking, let's say, it's a puzzle with the intracytoplasmic segment of these CAR. We have been adding CD28, 41BB, CD3 zeta, but let's do it in a kind of random fashion. So essentially, they have been collecting all the signaling motifs which are existing in the molecule which are used by T cells, B cells, even cells belonging to the myeloid lineage, mid-88 and so on. And so essentially they have been taking each of them, and they have been making combination of each of them. So by doing so, you start with gigantic number of signaling motif libraries, and they have been able to assess if some of these combination of domain were doing better than others. I mean, if you really look at face value, this literature that was published in really very high impact journals, there is no magic. I mean, no combination was in a way doing better than the one that were existing, but it's a very nice approach, and it will become even a better approach, because now the way people are trying to validate this new combination of motif that will then be used later on in CAR T cell to be used in clinics. The way they did the screening was by they have this library, they introduce it into a series of T cells, and they re-stimulate the polyclonal population of these T cells, which each of them is expressing one of these motifs, and they do stimulation again and again and again, and at the end they look at the one which have been winning, which are still at the end that survived this process of confrontation to the tumors. They look at what is in fact the combination of motif, and consider that is the best one. There is even a better way of doing a paper that came out in Nature last year, almost 1 year ago. It's very smart. What they did, I mean, one of the authors is really analyzing a lot of malignant T cells and identifying the translocation which might be driving the oncogenicity, or that may be passengers mutation helping to have these malignant T cells. And so they have been cloning each of these translocations, making a library of these different translocations which are present in malignant T cells, and introducing into CAR and transferring this CAR into a mouse. Here we have a screen which is going on in vivo, and the mice were having tumors, and after a couple of days or weeks, they are looking what are the T cells which are surviving the harsh environment on the tumors, and they end up on a translocation, which is very exciting, which confer a big advantage to CAR T cells. And the key point is that this translocation, the genes correspond, I mean, the two genes which are fused, they function only on antigen-dependent manner. So it means, fortunately, they are not oncogene by themselves, but we have to be extremely careful with all these approaches intending to enhance the durability and the proliferation of human CAR T cells, because sometimes, just took two examples published in the literature, it may lead to lymphoproliferative disorder or even to malignant transformation. So it has to be done in a very careful manner. As you are well aware, I mean, synthetic biology is really becoming also a very important tool in CAR T cell design. And for instance, that what is called now the fourth generation CAR or armored CAR, they are not only using a CAR to confer the specificity to kill the right targeted cells, but at the same time, when you engage the CAR, you manage to have the cells producing some cytokines that will contribute to boost the action of these CARs. And even more sophisticated approaches developed primarily by the group of Wendell Lim on the west coast of the USA, where he managed to make CAR T that are functioning only in some situations. For instance, it will allow them to deliver the effect of the therapy, for instance, to the brain, because you combine some kind of logical gate to have the CAR functioning only under some situation. And just to finish on that part also, as you know, so there was this original format that was designed by Zelig, and something which is very interesting is now we see more and more people which come back to the original architecture of the T cell receptors. Why? Is it because the T cell receptors are 10 to 1,000-fold more sensitive than the CAR to recognize antitumoral antigens? Which is very important, and they can really detect very few molecules when you compare them to the regular CAR. And amazingly, in 1987, I mean, well before Zelig-Eshar, there was a group in Japan, the group of Kuwana, who proposed this architecture of it. I mean, there was pre-hit or pre-star, where essentially you fuse the variable domain of an immunoglobulin on the constant domain of the T cell receptor. So I do believe that in the future there will be some move of the field toward this new format. But in a way, in the end, there is really a key question which is a question that was appearing already a while ago when people were testing combination of therapeutic antibodies or checkpoint inhibitor. Just to give an example, originally, for sure, I mean, we are having the CTLA4 and the PD-1 antibody as that were used as checkpoint. And then, as you are aware, I mean, a fair percentage of patient are benefiting of them, but unfortunately, most of them, they don't respond to anti-PD-1 or to anti-CTLA4. So the idea was to see if you can combine PD-1 with antibody X, Y, Z, and so on. And there is, in fact, so many antibodies that you can combine with PD-1 or CTLA4 that it turns out that it's a gigantic number of combinations, and that's very difficult to test each of these combinations because it's extremely costly in clinical setup. And in fact, it's even worse in the case of CAR T cells, but as you saw, I give you a kind of example of all the trials that people were doing for enhancing CAR T cells. So probably in the literature, I believe there is over 200 or 300 type of enhancement. So how to decide if a given combination of enhancement will be better than another one and will be tolerated, I mean, will be preferred to move to clinical studies. And we come back to an extremely important issue, which is the way in a preclinical setup, you will be trying to evaluate what is the best combination relatively to 100 other combination. It's not a simple issue, and there was a beautiful review that just came out at the end of 2024 in bioRxiv by a German group. It was systematically reviewing and doing a meta-analysis of all the preclinical models that were existing for CAR T-cell therapy and trying to figure out what was their predictive capacity for clinical outcomes. And as you will see, it's not completely perfect, so we do need to improve quite a bit these issues. So as you are aware, I mean, if we look at the present preclinical mouse model, which are used for evaluating CAR T cells, and it's kind of a consensus, it's not my view. If you look at this paper that was done by some people, I mean, the Society for Immunotherapy of Cancer, they were trying to list all the challenge that we are facing. One of the big challenge was, again, the nature of the preclinical mouse model that were used. And currently, what we are using, in fact, most of the time, we are xenografting tumors into immunodeficient mice. So it means that we are missing a lot of critical parameters. First, I mean, these mice, they don't have any myeloid and lymphoid T cells, so we completely negate their role. Same way, if there is an impact on the regulatory T cells, on the CAR, in the xenograft model we are using, we completely miss it. And something that we should not miss is that most of these immunodeficient mice, they don't have the proper lens node structure. So the way the CAR are homing and distributing is rather very shaky. So I think it's very important, for instance, it's even worse if you want to analyze fourth generation CAR, I mean armored CAR, where on the top of that, on the top of the CAR, you have the production of cytokines. The cytokines, as you are aware, they act on many cells which are absent in the immunodeficient mice. They can act on macrophages, NK cell, dendritic cell, regulatory T cells, and even on the endogenous T cells for boosting the effect of the CAR. So clearly, if you want to end up with meaningful information on the new generation of CAR that will be developed, you have to evaluate them in the context of functional immune system. So I just took two examples where people have just used some classical immunocompetent mouse model. And for instance, in that paper, they showed, they managed to show the importance of the CD40 ligand, which is expressed on these CAR T cells that has on the antigen presenting cells for boosting the response. And this other paper from a group in Switzerland, I mean, they use these immunocompetent mouse model to show the beneficial effect of IL-15 co-expression. For sure, if you do so, you have to go through one step where you will be using mouse CAR T cells. I mean, you cannot do it. I mean, it would be stupid to introduce human T cells in this fully mouse immunocompetent environment. And interestingly, also, there was a couple of papers which are popping out, which are using a humanized mouse model, for instance, for assessing a byproduct of CAR T cell treatment, which is cytokine release syndrome. As you are aware, when the CAR T cells are activated, they release a lot of cytokines that may be impacting on macrophage, that will be releasing themselves some pro-inflammatory macrophages that can create some dramatic effect. And so again, if you want to analyze the cytokine release syndrome, that is elicited by CAR T cell confronting to some tumors, you do need to do it in an environment that will comprise macrophages and for instance, endothelial cell that would be responding. And I was just choosing a very nice paper that came a couple of years ago by the group of Yvonne Chen. She was using humanized mice which are expressing human IL-3 and human CSF2. And it was very nice because she showed that it's a beautiful model of CRS. And on the top of that, it was very nice. She managed to add the CAR T cells producing a single chain LV which is neutralizing the production of cytokine, of inflammatory cytokines, and helping very much to fight these problems. And in that aspect, I mean, because we believe very much at JC Discovery that humanized and working on humanized and immunocompetent mouse models for evaluating the efficacy of CAR T cells. So we have been investing very much on some process, high-dimensional cytometry that allowed us in a single mice to essentially probe what is going on in many solid tissues, so where sometimes the action is going on, as well as the secondary lymphoid organ, and get a kind of global picture of what is going on in mice undergoing some tumor development, and not only using in the nose, but also using in many organs where the tumor may be metastasizing. So essentially, we for sure, I mean, we have all these tools which are available, and we like very much each time to discuss with our customers to fit on this knowledge that I was describing, the best experiment we can do for them. And we for sure also have some kind of roadmap. Where do we want to go to meet some of the present challenge of CAR T-cell therapy? And as you know, I mean, I was just listing a few of these challenges in the next couple of slides. So essentially, there's been a huge of improvement in the way of delivering the gene of interest into CAR. Through antiviruses or through knock-in, but we are still limited by the size of the payload that can be introduced into the antiviruses, for instance. And like many people, we are using third generation antiviruses. And as you know, I mean, they in between the two LTR, I mean, for sure there is the place where you put your gene of interest, but you also had to have them functioning at third generation lentiviral vectors. You have an endogenous promoter, and you have some sequences which already take some space in between the two LTR. And unfortunately, the space is limited. It's essentially 9 kb. So this means you can construct series of genes. If the genes are rather small, maybe you can fit 3 into that, but not more. I mean, it's impossible to go over. And I do believe that sometimes you will need more than 3 genes. It's a little bit the same situation from the work that was promoted by the group of Sadler and Ekem, where you knock in some CAR or eat at the level of the TRAC, the T-cell receptor alpha chain locus. And you do so to have some high efficiency. Generally, you bring the substrate, I mean, your template inside the T cells by using AAV vector, and AAV vector also they have a limit in size. So there is some kind of issue there, and I think we should go forward to meet the challenge which are ahead of us, and in terms of fighting solid tumors. And I was just, I mean, there is hundreds of reviews which are written on that to know what should we do to be able to tackle and make CAR T cells capable of eradicating solid tumors. I just list a few. I mean, and they were from a review for Carl Jones that were published very recently. So really, to challenge, we want to succeed in fulfilling. So essentially, we want to avoid to manage to manipulate CAR T cells, to having them avoiding the harsh avoiding harsh lymphodepleting chemotherapy. So this means in most of the clinical situation, before you treat with the CAR T cells, you have to subject the patients to some lymphodepleting chemotherapy, which is rather shaky for the patient. So it will be nice to develop CAR T cells which manage to engraft without that treatment. It would be nice also to enhance the migration of the CAR T cells from the blood if you infuse them, or from the draining lymph node if you try to do in vivo CAR T cells into the tumors via the bloodstreams. So there is beautiful data also which are in the process of being generating, where people try to engineer the CAR T cells in such a way that they are producing some enzymatic activity that will modify the extracellular matrix within the tumor micro environment and improve in a way the trafficking of the CAR T cells you are infusing inside the tumors. And as I was already mentioned, there is a huge amount of work which are intending to prevent CAR T cells to acquire some dysfunctional or exhausted state once in the tumor microenvironment. So for sure, you would like also to have them fighting regulatory T cells and conversely boosting intratumoral DC immunogenicity. You like to increase their metabolic fitness inside the tumor microenvironment. And also, as I was already mentioned, you would like to arm them with cytokines that will be also increasing their efficacy in the environment. On the top of that, it may be nice to remove a couple of checkpoint receptors to enhance again their potency, and also maybe to introduce some paper recently published by a group in Switzerland, the group of Pedro Romero, showing that you may need also to enhance the killing of tumor T cells, because many the tumors sometimes don't express the co-stimulatory ligand, so you need to pay specific receptor that will allow the CAR to kill in a better manner the cancer cells. And for sure, another big issue in solid tumors is to tackle the heterogeneity via the use of multi-specific CAR T cells. So essentially, that's the challenge that lie ahead of us. And so what we are dreaming is to develop some new vectors that will allow us to overcome these multifactorial challenges that are faced. So the point is that, I mean, just to go back to the slide I showed at the beginning, the CAR T cells or the T cells, which are fighting the tumors, they find big issues at the level of the tumor draining lymph node. I mean, they have to avoid the action of the regulatory T cells on the DC, for instance. At the level of the tumors, it's even worse. I mean, again, the Treg can be preventing the intratumoral DC to convert the TPEX into TX, and so you will be preventing these flows. So at the end, you remember the list I showed you in the previous slides. There is many challenges that CAR T cells or T cells are confronted with all along their life for fighting the tumors. And unfortunately, maybe I'm wrong. I wish I'm wrong, but I'm afraid that I'm not wrong. There will be not a single solution. Let's say you will do the miraculous experiment, you will change the CAR T cells, and all these challenges will be solved at once. So there is no one size fits all solution. And what we are thinking in a way is to build up some CAR that will express the multitask payload, essentially, that will be expressing in kind of successive manner a bunch of genes and under different promoter. For instance, we can imagine that the promoter will be very active in the tumor draining lymph node, and we'll need to express this green gene for meeting challenge 1, this one challenge 2, this one for challenge 3, and they will be operating at the right moment under promoter P1 in the tumor draining lymph node. But once they have been moving to the next stage, which is the tumors, maybe they don't need anymore the challenge have been met. So they will be facing some new challenges, and it will be very interesting at that time, once they reach the tumors, to under another promoter to trigger the right genes for fighting challenge 4, 5, and 6. So this means some very sophisticated engineering of CAR to at least fulfill the list of challenges I just show you in the previous slide. So I'm close to the end. As you know, I mean, CAR T cells, the field is now going way beyond cancer. I mean, there was a beautiful work of the group of George Chet in Germany demonstrating that, in fact, the CD19 CAR T cells that we are manufacturing for oncology can be reuse for fighting autoimmune disease. So that's something which is very exciting, and we are also inside GC Discovery developing a program around a mouse model that we have been developing that allowed to very rapidly assess the property of CAR T cells, which are intending to destroy some of the main actors of B-cell pathology. So that's my last slide. So I think that the field it's totally incredible. I mean, the field, the speed at which it is progressing. Unfortunately, there is considerable challenges, as you saw from one of my slides. We remain to unravel, and so we still need to probably know better the way T cells are functioning before translating them into clinical applications. So I think humility should be really very important reaction in front of that. But I really think that the prospects of integrating all these multidisciplinary approaches with advantages, and I tried to show some advantages of some preclinical model, but also drawbacks, and they are really excellent. And I do hope that inside this very complicated maze, I mean, we'll be able to progress and see the light. So thank you for your attention. Quentin, do you want to take over for the question?
Yeah, I will do so. And thank you very much, Bernard, also to share your thoughts on and predictions on the future of cell therapy in the cell therapy domain, transitioning from classical CARs to hit star formats to maybe TCR formats in the future. And also very excited to see what your team will do on developing and optimizing the model for autoimmunity and the use of CAR T cells in this model. So I'm eager to see the data and be able to share this with our partners and our prospects. Some of them are present today. So I will now ask everyone that has questions for Bernard related to his presentation to use the bubble with the question mark logo in on the GoToWebinar program, and yeah, hit us with some questions. If we're unable to answer them today, Bernard will follow up by mail. Also, so it's clear for everyone, everyone will have the recordings of this webinar as well as a certificate of attendance. So that's will be sent by my colleagues from marketing. But I will give you another minute to send you some questions. I see them coming. So maybe I will start with some questions. Okay. So one question, very specific question, Bernard, on the type of mice strains to use to check the efficacy of CAR T cells, and what are the parameters that need to be studied, I guess, within the efficacy study to really say, okay, the CAR T cell works.
I mean, it's unfortunately there is not a single answer to that. So as you saw, I was taking an example a little bit earlier of the use of humanized mice for CRS. I mean, for documenting these very harsh condition that was resulting for the treatment, and probably the mice I was describing, GM-CSF, CSF2, IL-3 are very convenient. I mean, you can really extract some data that will probably be extremely strong to be translated to the preclinical situation. For the other, for the question of the CAR T efficacy, for sure, you will like to have the best humanized mice, which have been properly reconstituted, where you will have the myeloid component, the lymphoid component, and so, as you know, I mean, inside in our group, there is a lot of effort to manufacture this kind of perfect mice and to avoid variability between the individual of a given batches. So there is a lot of models which are really already available. Then there is another issue, let's say, which is, for instance, we are using very much in the lab some adenocarcinoma, at least for the mouse immunocompetent model, MC38. But the way we deliver it to the mice is by doing intradermal immunization. So it's not the right position. So if you want to go further, it will be, I mean, for me, the best would be to combine the top model of humanized mice with orthotopic engraftment of the tumors. In such way, you will be really very close to the situation. There is another issue which is very important, is that, as you are aware, but it's very complicated to work with. I mean, we have been in my former academic lab. One group has been working with a group in Strasbourg on some adenocarcinoma of the prostate, and so we wanted the perfect model, and the perfect model was a model in which we don't graft the tumors, but we trigger by genetic tool the development of the tumor from scratch. The problem is that the experiment, they last months because the tumors develop very slowly. So at the end, it's a kind of an equilibrium, because you want the result as rapidly as possible, but you don't want to do too much quick and dirty experiment. You find you always try to find a kind of an equilibrium in between quick and dirty and the perfect experiment, because it's like that. So I'm very sorry. It's a very complicated answer that I provided, but I think it reflects the reality on the ground, and the importance of discuss each time with the people to really know what they want to measure, what they want to extract from the data, and to try to provide the best solution.
Thank you very much, Bernard. I hope this answers your question. Another question related to the use of CAR T cell treatment, I guess, in the clinic. The question goes as follows: is the bottleneck of CAR T cell treatment the cost of it? So the cost related to the CAR T cell treatment, is this the bottleneck
It, it, it,
in?
it is one bottleneck, but as you saw from one of my slides, there is also a huge amount of still scientific bottleneck in terms of, you want to fight solid tumors. So I think you have probably, you have to add to pile up to these money-wise bottleneck all the other scientific bottleneck which need to be, I mean, there is a lot of progress, but still, I mean, for instance, the TIL, which are the tumor infiltrating lymphocytes, which people were thinking, I mean, it was the group at NIH were starting 20 years ago to try, maybe more, to try to develop them. And at one moment, people were very reluctant, it will go nowhere. And very recently, I mean, there was very interesting reports coming from this group at NIH with a company that is matched to them, and also with the people in Netherlands, where the TIL are coming back, because in fact they're resolving one of the very key issue of the solid tumors, which is the nature of the antigen. So the TIL, they do the business themselves. They choose, we don't know exactly well, but how they react to neoantigens. But when you manufacture them from scratch, you have to be very careful in terms of the way you arm these T cells, which are directed against solid tumors, to avoid, if they say there is parenchymal cells, which are expressing tiny level of that, to be also afflicted by the CAR T cell treatment. So I think, I mean, I think if we had a very good, it go back to my part of my talk, if we have a very good preclinical model that will allow us to sort out in a way, if I should use this set of enhancement of the CAR versus 200 other one, it will also probably short the money we have to invest in the development, and probably at the end also shortcut the cost of the at the clinic. But I'm not a clinician, so I'm pretty badly located to answer this question.
Okay. Thank you very much, Bernard. Then another question, a very interesting question. What's your view on reprogramming immune cells directly in the body to give rise to in vivo CAR T cells, albeit in cancer or autoimmunity.
Yeah, that's really, I mean, for sure. I mean, that's a very exciting perspective, because in a way it will shortcut the cost of the manufacturing of the car, because essentially you will provide the piece of the puzzle to your own body to do the manufacturing inside. As you know, there is a lot of company and academic labs which are working on that. I think there was a major boost to these view, based on which is far simpler, but at least you know all the lipidic vesicles or the nanoparticles that were used for packaging the RNA for SARS-CoV-2, for instance, for allowing us to have some very successful vaccine in a few couple of months. So it's part of this technology for unpackaging RNA, are also work on the side of the vaccination. So I think if you move into the CAR-T business, all this knowledge which has been accumulated around SARS-CoV-2 vaccination, that will go further. For sure, the challenges are enormous because you do need to find the proper way of targeting, let's say, the viral particles or the nanoparticles that you will be delivering in vivo in the patient. So you need to target the right cells, but then you need to deliver in a very efficient manner the payload that you want to deliver need to be expressed in a proper manner and for as long as possible. But no, it's a fascinating it's a fascinating domain, and I'm sure in the next couple of months or years, I mean, we'll see a major development in that field. Now that's a kind of a growl, because it will fix so many of the issues that we want to fix.
Okay, thank you, Bernard, and let's hope indeed that this could circumvent, I would say, the costs, like briefly mentioned, by
I
applying
just want to
directly in
so
vivo.
I just want to add something because
Yeah, yeah, go
I
ahead.
know I know that I'm T-cell obsessed. I mean, I've been working on T-cells for 40 years of my life, but I've been also working on dendritic cells and macrophages. And I do know also there is NK cells. I mean, I'm just joking. So there is not and gamma delta T cells. So for sure, I mean, I've been very much focusing on alpha beta T cells. But I mean, all these other type of T cells, I mean, including May T cells or other unconventional T cells, maybe they will help us also to fight cancer, including for sure NK CAR or macrophage CAR or so on. So just to say, to apologize for those people which are very much working on that. I think they're extremely important. Just for the sake of time, I was focusing on alpha beta T cells.
Yeah. Okay, thank you, Bernard. So, with the compliments to your great talk as well, uh, I guess the question is related to the heterogeneity of tumors and how you assess this heterogeneity, I guess, during the engineering of CAR or TCRs.
Yes, that's a very so it's a question that was already popping out for hematological tumors, where when you put a selection pressure, let's say you will have a CAR with CD19, you tend to select cells inside the tumors that will not be expressing CD19. So what people are doing, I know the group of Stan Riddle in the US and many other groups, they are trying to manufacture CAR T cells, which will be expressing for sure the CD19 specific CAR, but also a CAR specific for another antigen expressed by the hematological malignancy. So in such way, it's a question of probability. I mean, the probability to escape to 1 will be 10 to the minus 3. The ability to escape to 2 will be the product of 2. So you may end up on the safe side. It's even more complicated for the solid tumors. And there is an unbelievable amount of work which is done at the world level on how to define the best antigen for solid tumors. For sure, the The neoantigen, they are the best. I mean, these mean mutation which are specific to given tumors. The problem is that they are not universal. There is also the beautiful work done by many companies on finding some splicing defect or open reading frame which are only used in the tumoral cells and giving rise to peptides which are unique to the tumors. So you may try to use these molecules. And then I was speaking also very much in favor of the T cell receptor, which because they're extremely potent, they will be capable of recognizing tumors expressing very small amount of a tumor antigen. The problem is that if it turns out that you have also normal tissue which is expressing a small amount of that, they will be also subjected to the killing by the CAR. So again, it's always the same. I mean, it's a kind of an equilibrium. Where do you put the cursors in between that? But no, sure. I mean, that's a very important domain. And there was recently several groups manufacturing some brand new. It's neither immunoglobulin nor T cell receptor. It's brand new folding modules that they managed to have them recognizing essentially the peptide. They become very peptide centric without caring too much about the alpha helices of the HLA. So you will end up with kind of intermediate between T cell receptors and immunoglobulins, and they may be also contributing to solving these very important problems of the heterogeneity in the antigen of the solid tumors.
Okay, it's clear that specificity will be very important, Bernard. Another question, rather interesting, and it's related to the complexity of antimicrobial resistance currently. What's your view on the use of CAR T cells for bacterial infections?
I am not. I mean, with some of my colleagues at one moment who were thinking to embark into that domain. So I don't know much. I was reading reviews in the literature on that, so I don't have any first-hand experiment. I still think it's a kind of complicated, and also it's a question of the balance of the risk and the benefit. So for sure, I mean, unfortunately, when people are about to die of cancer, you can use ARCH treatment. Let's say you will do this lymphodepletion regimen because there is not so much solution to get rid of that. If I take not the example of bacterial infection, but of autoimmunity, it's clear that autoimmunity, it's a very bad disease, but sometimes it will not be lethal in short term. So will you take the risk to treat this autoimmune patient with some lymphodepleting, harsh lymphodepleting? Probably not. And so for the bacteria, I don't know if this risk will not be also too important to reach a final result. But again, I prefer to shut my mouth because I know nothing about that.
If they ask your view on it, you have to share your view on it, and it's for me a first. I never currently know. I don't know any companies that is adventuring in this field yet, so will be interesting to see in the future. Um, okay, so a last question until now, so that will fit with our timing schedule. Um, is it possible to induce stemness or naive-like phenotype directly from transient CAR stimulation in multispecific CAR T cells?
One of the slides I was mentioning where the people were claiming that they have some eternal, I mean, some CAR T cells that were functioning forever. That was exactly their what they were intending to do, to find genetic modification that will make them capable of growing forever without becoming malignant T cells. And this paper that was published in Journal of Experimental Medicine was quite impressive. And there was a recent paper published in Science where they find the transcription factor called KLF2, which is in a way pushing in chronic antigenic stimulation the T cell in the right way. So this means they don't go anymore into some kind of exhausted phenotype, but they go in a more classical effector memory pathway. So there is a lot of, again, I'm not an expert at all of that, but there is a lot of group which are trying to manage to change this kind of stereotyped behavior of T cells in chronic situation to avoid having them becoming dysfunctional. And it's really hardcore molecular biology and transcription factor business.
Okay, and now the questions are coming in, Bernard. So let's keep it interactive and continue. So also very interesting question in my opinion. So they recently observed that tumor infiltrating, I guess, immune cells with high avidity for their target were largely ineffective and exhausted compared to their less avid counterparts. So I guess TCRs that are less with less
It's,
affinity.
yeah,
Do
yeah.
you think this is also an issue to watch out for in CAR T cell therapies?
Sure. I mean, that's already the point. I mean, we are not always in a black and white situation, and probably if you know all these, if we go back to just regular T cell physiology, uh, why do you have all these co-inhibitors? It's just because if you stimulate too much, you create a lot of problem. You create problem in the tissue because you will be killing a lot of cells which should not be killed. But something which is very interesting is that some people, when they were creating mice, where they were ablating PD-1 when the T cells were born, and they thought that it will be better for them, that they will be surviving more. But in fact, they are burnout. I mean, they are too much stimulated, they receive too much signals, and they don't work anymore in the long term. So probably the most is not the best. So again, and I know it's a very interesting and very important question. Just the view to find the right regimen that will not burn, they will not become burnout, but they will be very efficient and not too slippery. So it's again, I go back to unfortunately, if everything was black and white, biology will be very simple. But when you look at all these many co-inhibitory receptor and co-stimulatory receptors which are present at the surface of a T cells, they are therefore something, and we don't understand much what they are doing all together. I mean, we have some glimpse of what they are doing. So at the end, I think once we know how the T cells or the CAR T cells are managing to compute all this information, and if it do good or bad for them, then probably we'll be able to be more rational in the way we do presently.
So affinity of your binding moiety is very important to assess and to include, I guess, in the selection funnels of your
But
CAR
look, it's
construct.
always it's very complicated because we like to have the maximum affinity for grabbing small tumor cell line expressing tiny amount of stuff. But if it hurt your T cells, it will be useless because essentially you will embark them into a death pathway.
Okay, I think with this we can conclude on this very first webinar of JC Discovery, which I'm very proud to moderate with the help of Javier and of course Bernard. Thank you very much for your sharing your knowledge to this community, and let's hope we have an occasion in the future to reconvene on this and to maybe give an update to everyone on your predictions and your view on the future in
Yes.
this exciting field. So yeah, thank you everyone for joining again.
Thank you.
I wish you a very pleasant afternoon or good morning, depending on where you come from. So yeah, uh, let's
Bye
reconvene
bye.
soon. Thank you. Bye.